folks. We are going to talk about smooth muscle today. As a recap, the last 2 weeks we talked about skeletal muscle. Skeletal muscle is muscle that you can voluntarily control. So you think of muscles you've been learning in anatomy, your bicep brachii, your tricep brachii, your brachialis, all the muscles in your neck and your head that use control posture and your back. So all those muscles that you can control, those are voluntary skeletal muscles. Smooth muscle is involuntary, so you cannot directly affect how these muscles contract, although they will respond to the environmental stimuli and to signals from the nervous system. So to get started, we'll start with some announcements and then we'll go into our topic. So again, we have our Blackboard quiz this Friday, available at 4 p.m. Eastern Standard Time. Normal format, 20 questions, multiple choice, a few true and false, and you have 30 minutes, says 20 minutes. You have 30 minutes to complete, and there will be unlimited attempts. Now, as a reminder, quizzes in total account for 10% of the final grade, but also they seem to help more in the sense of, for the past 3 years, I've analyzed midterm performance and final exam performance. Those students who persist at repeated attempts in the quizzes until they get at least 80%, do better, significantly better in the midterm and the final, because it's the repeated exposure to the information, it's seeing the style of questions that are going to be asked, and also just an objective measure of how well you know the information. Okay, and in terms of this quiz, it will include new information, but also be some information from other quizzes also. And also, we have a midterm next week. So let's talk about an exam review. So for your midterm, it will take place on Tuesday, October 19th, 2021. That is 1 week from today. The start time will be 2 p.m. and that is Eastern Standard Time. So make certain that your calendar, your clock, whatever you're using is set so that you are synchronized with 2 p.m. Eastern Standard Time. And the location will be on Blackboard in the assignments and tests. So here's how it works. I'll be logged onto Blackboard. You should get logged onto Blackboard by 1:50 p.m. Eastern Standard Time. You complete the exam, there's a midterm exam and then a turn it in component. And then when you're finished, you log off, you smile because you studied, you did the quizzes, you did great. Couple of notes, of course, it is different in ensuring academic integrity online as opposed to in-person exams. So it's my request that you adhere to the principles of academic integrity. What does this mean? This means you perform the assessment yourself. You're not using resources while taking the exam. You're not collaborating with other students or other people, and you're not copying material from the exam in any manner or fashion. In terms of the policing and enforcing of this, I reserve the right to review the data analytics for each student's exam. So we can look at different bits and pieces of information about your exam performance that will give us an indication of, this looks a bit suspicious. Let's look into it deeper. If we notice a discrepancy, it is my decision to investigate the matter further or not. And you need to be aware that let's say you're a bit naughty, and let's say that you take answers off the internet, copy and paste, or you paid someone else to do the exam for you, and we notice this in the data analytics, you may get charged with academic misconduct. In terms of following up, we could even set a one-on-one meeting where I will ask you pointed questions about your exam and performance on your exam and decide from there. And this is given a history. We now have over 1.5 years of administering exams online, so we're getting more and more efficient at this. So yeah, I just want to make sure that we are rewarding the students who abide by the rules. In terms of technical issues, there's over 100 of you, so at least 2 or 3 of you will have some technical issues. If you experience difficulty during the exam, you need to contact me right away. And if you don't, it's 0 in the evaluation. So you cannot come to me after the exam and say, well, I couldn't get logged in, I had problems with my internet, whatever the case may be. That is my email address, matt.kohler@georgiancollege.ca, or also I'll be in the WebEx room. Now, if it takes me 2 or 3 minutes to respond to your email, keep in mind I may be dealing with other students, but I will be logged on during your evaluation also. One thing to note also, we can't always control our internet access. So if you are renting and the landlord is providing you with internet, maybe talk to them and say, listen, just want to ensure or be sure that I have internet access during this day and time. And also, I mean, for example, we still have students who are in countries, international countries. And one example is in one country, there was a province-wide exam for people to become public school teachers. And during the exam, the province cut out the internet for all 25 million residents. So make sure you're not in that situation also, because you will receive notification of that. So just maybe do some probing and questions right now to ensure that you have access to the internet. In terms of missing an exam, if you miss an exam, in order to be given a makeup exam, you need to contact me before the start of the exam, and you need to bring official documentation to myself. And I warn students, I say, listen, if you have a family tragedy, someone's passed away, I'm going to require something. So you need to give me something in terms of official documentation. The opportunity to write a makeup exam is my decision, and my decision alone. So I will base it off of when you contacted me and what documentation you brought forward and the nature of that. For the exam, what are you allowed? Well, you need some type of computer, whether it's a tablet, laptop, or desktop. I would recommend use a desktop. Don't do the exam on your phone. That's the most awkward. Keyboard, internet connection, and of course that great big healthy brain of yours. In terms of the material and the topics, it's everything we've covered so far. So we went over cellular organization in weeks 1 and 2. Week 3, we went over cellular metabolism. In week 4 and 5, we went through skeletal muscle physiology, and this week we're going to go through smooth muscle physiology. So anything covered in the class or posted in the textbook readings should be studied and reviewed. The format will be, there will be multiple choice questions or multiple guess, depending on how well you know the information. There will be images, and so it may be, here's a list of words, and which word belongs on A, which word belongs on B. We'll have ordering or matching questions also, and then there will be a long answer question. Each individual exam will consist of questions randomly drawn from a pool of questions. So no two students will have the same exam, but it will be the same degree of difficulty. In terms of the long answer question or the essay question, there will be the final, so you'll go through the multiple choice, true, false, and then the images and the matching and the ordering. And then the last question will be, this is the last question on this part of the exam. It is not worth any grades. I acknowledge that when I submit this question, a Word document will be revealed to me in assignments and tests. I am to download that document, complete the answer, and then submit it to turnitin.com. So that would be in the assignments section, assignments and tests, and Turnitin assignment. And the description will be posted as a file. So when you download that file, you will basically see what the instructions are for that question. All the answers are run through turnitin.com, so don't copy from other sources, and don't word swap or paraphrase, right? Because we want basically your answer and as your interpretation of the information. In terms of study tips, so what do I recommend? First off, review the notes from class and also use the textbook readings and read through the textbook and also use the textbook to supplement the information from the notes in class. So if you don't understand something, maybe you're still not clear on the electron transport chain from cellular metabolism. Maybe you're not really clear on how exactly a skeletal muscle contracts. Go into the textbook and read through the details because the textbook is there to help you. And that's where we draw our information from. Learn the broader information first and then the smaller details. So an example is for cellular organization, for the different parts of a cell, learn what they are called first and then learn what they do. Test yourself, write out your notes and then do it again and again and again. And when I say write out your notes, don't just write out word for word what they are, but write as if you understand what they are, like as in your own words. And then review the online quizzes, and I would say start now. And then if you do have any questions, feel free to email me. If we need to, we can set up an individual meeting and then we can go from there. Okay, and also as a note, the recordings of all these lectures are posted until 11:59 p.m. Eastern Standard Time, the Friday following the lecture, and then they come down. So that is basically your exam review. For labs this week and for next week, I will be in this WebEx room. So it'll be an opportunity. If you have questions, you know that I will be in this WebEx room. We're going to treat our lab time as basically an open question time. So if you have questions or you need me to explain something or you require clarification, then certainly we can meet in our lab times and you can come to any of the lab times and we'll be able to answer those questions. Okay. So the smooth muscle, this information is found in chapter 9, pages 312 to 317. So it's basically 5 questions, but what are smooth muscles? Well, smooth muscles, as we said, are basically the involuntary muscles of the human body. And what does that mean, involuntary? Well, it means that basically you cannot directly control how they contract. They're found in the walls of most hollow organs. So any organs, any structures that have a space inside, with one exception, the heart. The heart does not have smooth muscle around it. So in this case, we see on the left-hand side a picture of someone's digestive tract. So that's the liver at the top, the stomach underneath it with your gallbladder, which is in yellow, or no, sorry, your pancreas, which is yellow, and your spleen, which is purple on the right-hand side, and your gallbladder, which is green underneath the liver. And then you see the large intestine and the small intestine, and part of the small intestine is pulled out and is box through it that shows it's a cross section. And then we see a cross section of the small intestine in the middle picture. And we see that there are 2 layers of muscles, of smooth muscle around the small intestine. So they are spindle-shaped fibers, right? And they are thin and short. They have 1 nucleus per cell, and there are no striations. So with skeletal muscle, it had those stripes. There are no stripes. And so if you see on the right-hand side, you can see the longitudinal layer of the smooth muscles and the circular layer of the smooth muscles. Also, there's no connective tissue except the endomysium only. Okay, so we don't have the paramecium, epimecium, we only have the endomysium. And when we're looking at this diagram, we can basically see that we have the circular layer, which goes around the small intestine, and the longitudinal layer, which goes basically along the long axis of the small intestine. And there's a reason and there's a function behind that. In terms of the microscopic structure, let's bring it down a bit. So most blood vessels contain smooth muscle. And so in actuality, you're aware of this because when you are out and somebody says something embarrassing, or you see someone that you are attracted to and they walk in and one of your friends says, oh, I think they're going to talk to you, and you blush, those are the smooth muscles around those blood vessels relaxing and opening up those smooth blood vessels, and your face goes red because why? You have more blood going through there. So there's 2 layers, as we mentioned before, and these are opposing fiber sheets, if you will. You have first off your longitudinal layer, not longitudinal, longitudinal, right? And so this is parallel to the long axis of the organ. So this, if we're thinking about this image right here of the small intestine, we can see that basically the longitudinal layer runs this way. Okay. The other layer is circular layer. And this runs around the circumference, almost fit in there, of the organ. So in this case, the circular layer basically goes around the circumference of this. So when the longitudinal layer, so remember, the only thing a muscle can do is contract. It cannot elongate itself. So when the longitudinal layer contracts, then that's when we see that it shortens. Okay, so these muscles contract and it gets shorter. So imagine if you look at my camera, if this mailing label, this tube, was part of your small intestine, the small intestine would get shorter when those longitudinal muscles contract. The circular layer, when they contract, they constrict the inner space, what we call the lumen, L-U-M-E-N. So they constrict the inner space. Well, both of these combined the combination of contractions of the two layers leads to peristalsis. Okay, peristalsis is basically it's an alternating contractions and relaxations. And this leads to mixing and squeezing substances forward. So this is what causes the food as it's being digested to be passed through the small intestine, that it squeezes and then shortens, and then squeezes and then shortens, and pushes the food along this tube. In terms of also the microscopic layer, there's no neuromuscular junction. So remember before with skeletal muscle, we had the neuron coming down and would meet with the muscle fiber. You had the little gap, the synaptic gap, and then the action potential, the signal would come along the neuron. It would cause acetylcholine to go across that gap and then cause an action potential in the muscle fiber, which would then cause contraction. Well, we don't have that junction here. Instead, we basically just have these, what we call autonomic nerve fibers, right? So these are automatic and they're arranged as varicosities, which are really bulbous swellings of nerve fibers. Okay, so they store and release neurotransmitters, and there is a wide synaptic cleft, which is known as a diffuse junction. Okay, and we can see a little bit of a close-up here where we have these synaptic vesicles and the mitochondria in there and these varicosities. So they release the neurotransmitters across this diffuse junction. And these bulbs are all spread across the different smooth muscle fibers. In terms of also the microscopic structure, we see that this at the top is a relaxed smooth muscle fiber, and this at the bottom is contracted smooth muscle fiber. So what are we seeing here? Well, smooth muscle fibers do not contain sarcomeres, right? We don't have those as organized myosin actin sarcomeres, like those individual sarcomeres. They don't contain myofibrils, and they also do not contain T-tubules. The sarcoplasmic reticulum is less developed, than in, say, the skeletal muscle with most calcium. So when we say Ca++, we mean most calcium for contractions coming from extracellular sources. So the calcium is coming from actually from outside the cell. So the difference is the sarcoplasmic reticulum inside the skeletal muscle has the calcium. So the calcium is inside the cell, but it's inside the sarcoplasmic reticulum, separated from the myosin and actin. Well, in this case, the calcium in a smooth muscle fiber is coming from outside of the cell. And you have these things called caveolae, which are pouch-like enfoldings in the sarcolemma. And you can see these caveoli as the little gaps on the top picture. Well, what do these do? Well, these have calcium channels, and they open to permit rapid influx of extracellular calcium. So these little caveoli, there, and there, that's where your calcium channels are. So the calcium is coming from outside of the cell into the cell to bind with the troponin to move the tropomyosin out of the way, but the contraction sequence is different. There are some similarities, but there are some differences from skeletal muscle. Also, in terms of the microscopic structure, the thick filaments are fewer than in skeletal muscle, and there are myosin heads along the entire length, right? So the thick to thin ratio, so the actin, or sorry, I should say myosin to actin is 1 to every 13. Whereas before we had 1 myosin for 6 actin, now we have 1 myosin for 13 actin, right? So this is lower than skeletal muscle. And really the ratio when it worked out was a 1 to 2 ratio in skeletal muscle, right? So for every myosin you had 2 actin, here for every myosin you have 13 actin, right? The thick filaments, so the myosin, have myosin heads along the entire length, right? And smooth muscle is, as powerful as skeletal muscle. So when it contracts, it can create as much force as skeletal muscle. Now, there is no troponin complex. There is tropomyosin, but no troponin. And so we have a protein, calmodulin, which binds to calcium. And we'll go through the contraction process in a second. In this image, what we're seeing, this is an electron microscope image of the parts of the smooth muscle cell in a transverse section. And so we can see that we have the dense bodies, which we'll explain a little bit. The dense body right here. We have mitochondria. So there's a mitochondria right there. We have caveolae, right? Those where the calcium channels are. And then we also have these intercellular junctions. So there's a circle right there with an intercellular junction. So we will have thick and thin filaments arranged diagonally, okay? Which means your myofilaments are spirally arranged And when it contracts in a corkscrew manner. So when the muscle contracts, it's like twisting when it contracts. And then you have these intermediate filament dense body network. Okay, so what does this mean? They're kind of like on a lattice, right? So this dense body network goes over the surface of the cell. So we go back, we can see that the dense bodies are here, and we have these intermediate filaments which are joining them together. Okay, so it's a lattice-like arrangement. Okay, and you have those non-contractile intermediate filaments. And what these do is they resist tension. Okay, so these dense bodies are proteins anchored, proteins that anchor filaments to the sarcolemma at regular intervals. Okay, so we see a relaxed muscle fiber up here, contracted, and these dense bodies and these intermediate filaments basically bind around the cell. Okay, it's similar to the Z discs of skeletal muscle. So during contraction, areas of the sarcolemma between dense bodies will bulge outwardly, and they make the muscle cell look puffy, as you can see right here at the bottom. So we see a muscle cell that is, see muscle cell here is relaxed. We see that one's contracted, and you can see that it looks puffy. It's bulging out because we have those non-contractile intermediate filaments. It means these are, it's almost like rope that doesn't contract, and the dense body are anchors. So think that it's like someone wrapped this rope around the cell and then anchored it to the cell with these dense bodies. So when the cell contracts, it's bulging in between these little rope, in between the intermediate filaments. So where students sometimes get confused is, you want to be able to have a quick reference in terms of, well, can I compare the different types of muscle fibers? So we have 3 types of muscle fibers. We have skeletal, which we covered the last 2 weeks. We have smooth, which we're talking about today. And then after the midterm, we're going to talk about cardiac, because they're all significant types of muscle fibers. Now, if you're going into the fitness and health promotion field, or even if you go on into something such as physiotherapy, occupational therapy, kinesiology, massage therapy, what athletic therapy, you're dealing primarily with skeletal and cardiac muscle cells. Now, skeletal directly in terms of performing exercises and movements that will help with functioning and balance of skeletal muscle cells, and then cardiovascular and high intensity exercise that will help, the movements and rehabs with skeletal, high intensity cardiovascular aerobic type work for cardiac. We can't really do anything, or we don't really do anything directly with smooth muscle cells. It's not as if we give exercises or rehabilitation for smooth muscle cells, but it can receive benefits indirectly by working on skeletal and cardiac muscle cell. And so a good example is that people who participate in regular physical activity have a much lower rate of colon cancer, colorectal cancer. And why is that? Well, that's because that the movement will assist in regular digestive processes, and that will help, to reduce the risk of colorectal cancer. Okay, so we don't work on smooth muscle directly, but we can work on it indirectly when we are doing exercises and rehabilitation. Okay, so in this chart, we're basically looking at body location and the cell shape and appearance. Well, skeletal muscles are attached to the bones or fascia, that connective tissue, or to the skin. They're single, they're very long, multinucleated, obvious striations. Well, smooth muscle here, any walls of hollow organs with the exception of the heart. And they're single, uninucleate, like fusiform, like, distinct shape, no striations here. In terms of the connective tissue, well, we talked about how skeletal muscle has epimysium, paramysium, endomysium, but we only have the endomysium in smooth muscle tissue. And we're not covering skeletal or cardiac right now, so we won't worry about it. Are there myofibrils composed of sarcomeres? Yes, in skeletal muscle. No in smooth muscle, but we still have actin and myosin. Are there T-tubules? Yes, in skeletal muscle. No in smooth muscle. Do we have an elaborate, like a complex sarcoplasmic reticulum in terms of this is where calcium is stored? Well, yes, in skeletal muscle. That's where your calcium comes from. So again, try not to learn facts in isolation, but think about why is it that skeletal muscle has a more complicated sarcoplasmic reticulum? Well, because that's the source for calcium. That's where calcium gets released and where it gets taken up again. Well, in smooth muscle, no, not really, because why? The calcium comes from outside of the cell. Do we have gap junctions? Well, what are gap junctions? Think of gap junctions similar to doorways between two cells. So the two cells are connected, has a doorway, which is a gap junction. We don't have this in skeletal muscle, but we do in some smooth muscle cells. Do we have individual neuromuscular junctions? So one neuron comes down and goes to individual muscle fibers. Yes, in skeletal muscle. No, not in smooth muscle. And then the regulation of contraction, like what controls the contraction of these muscles? Well, in skeletal muscles, it's the nervous signal. It makes sense because you think, I want to reach for that cup, or I want to reach for that plate, or for that pen. So it's the nervous system. Well, in smooth muscle, we don't control it. So it's autonomic, like it's automatic nerves. Could be hormones, could be chemicals, could be stretch, like a stretch contraction reflex. Source of calcium for the calcium pulse, right? Calcium gets released to start the contraction. The skeletal muscle, well, it has a complex sarcoplasmic reticulum, so it must be in the sarcoplasmic reticulum. Smooth muscles, a little bit from sarcoplasmic reticulum, but most of it's from outside the cell. The site of calcium regulation, so what does calcium bind to in order to initiate contraction? Well, it's troponin in skeletal muscle, and it's a protein called calmodulin in smooth muscle, so there's a difference there. Pacemaker cells, which basically automate the contraction of the muscle fibers. Well, we don't have this in skeletal muscles, but we do have it in some muscle fibers. What happens when the nervous system signals a muscle fiber? Well, in skeletal muscle, they get excited, they contract. In smooth muscle, they might contract or they might be inhibited from contracting, depends on the nervous system. Speed of contraction, so how fast do these muscles contract? They're slow to fast in skeletal muscle, so we have a range of speed contractions. In smooth muscle, it's very slow. There's a rhythmic contraction. No, not in skeletal muscle, which we don't want. But yeah, in the smooth muscle, well, think about why would you want rhythm, like regular repeated contraction? Well, think about the small intestine. You have the circular and then you have the longitudinal fibers contracting in rhythm to help propel the food along that you're digesting. What's the response to stretch? Well, in a skeletal muscle, the amount of tension can be produced, can increase to a degree of stretch to a certain point. With a response to stretch in smooth muscles, they can stretch and then relax. And we'll get into that in a little bit. Metabolism could be aerobic or anaerobic with skeletal muscle, right? You might need to sprint, you might need to jog, you might need to walk. With smooth muscle fibers, mostly aerobic. So in terms of the mechanism of contraction with smooth muscle cell fibers, we are going to have slow, synchronized contractions. They're not fast, and synchronized, there's a rhythm to it. The cells are electrically coupled by gap junctions. So it's like these doorways in between the cells. Okay, and why is that? Well, the action potential can be transmitted from fiber to fiber, which we want this, right? You might want the longitudinal fibers here to contract, and the longitudinal fibers here to contract, and the longitudinal fibers here to contract. Why? Because we're pushing food stuff down the intestine. Some cells are self-excitatory. That means that they depolarize without external stimuli. So you don't need to have a signal. They're just like, contract now, contract. That's what they do. They don't need someone else to tell them what they do. But what these cells like can self-contract do is that they will act as pacemaker cells for sheets of muscle. So it'll kind of be like they're the boss, and they will contract and get the rest of the cells around it to contract. The rate and intensity of contraction is modified by neural and chemical stimuli. So what does this mean? The nervous system will affect how the contractions occur, but also chemicals. So chemicals within the environment can affect how well these smooth muscles contract. So the mechanisms for contraction, okay? So this is a diagram, page 312 of your textbook, and we'll just go through the top and then we'll go a little bit more in depth. Calcium will come in from the extracellular fluid, and the calcium ions enter the cytosol, that's the inside of the cell, from the ECF, extracellular fluid, through a voltage-gated or non-voltage-gated calcium channel. Or you might get a little bit from the sarcoplasmic reticulum. Calcium binds to and activates calmodulin. Calmodulin activates the myosin light chain kinase enzymes. The activated kinase enzymes catalyze the transfer. They help the transfer of phosphate to myosin. So basically, when this calmodulin is activated, it activates the kinase, which then helps transfer energy to the myosin head. And then the myosin basically form the cross bridge and they start to contract and shorten. So you still have actin and myosin. They're still your two main players, if you will, two main proteins, right? Which interact by the sliding filament mechanism. Okay. And the final trigger, the final signal is increase intracellular calcium, right? The ATP, ATP energizes, that is right. Energizes the sliding process. And then contraction stops when calcium is no longer available, okay? So we have some differences. You still get some calcium from the sarcoplasmic reticulum, but most from, but most is from extracellular fluid. Calcium binds to calmodulin and not troponin. And the activated calmodulin then activates myosin kinase, right? It's a myosin light chain kinase. Myosin kinase phosphorylates, right? So it breaks apart the ATP and gives some of the energy to the myosin head. Okay, and this leads to cross bridge formation with actin. And then you have your contraction that starts. Stopping it is a bit more involved, okay? So it's not just a matter of sucking up the calcium to the sarcoplasmic reticulum. And so it's more involved than in skeletal muscle. So the calcium detaches from the calmodulin. And then the active transport of calcium into the sarcoplasmic reticulum and out of the cell. And we have dephosphorylation of myosin to inactive myosin. So calcium comes in, binds calmodulin. Calmodulin activates the kinase, the light chain myosin kinase. That allows ATP to transfer energy, phosphorylate the myosin head, which gets activated, contraction starts. Stop contraction, calcium goes off of calmodulin. We actively get it out of the cell or into the sarcoplasmic reticulum, and then the myosin becomes inactive. So in terms of some of the properties, it is slower to contract and relax, okay, than skeletal muscle. So it will not happen as quick, okay? It maintains contraction for prolonged periods. So whereas skeletal muscle will basically tire out if it's maintaining a contraction, smooth muscle tissue doesn't do that as much. There's little energy costs. So it's very efficient at contracting. It has slower ATPase. This is the enzyme. So this is the enzyme that breaks apart the ATP. It's slower to act. And the myofilaments may actually latch together to save energy. So the myosin can actually latch onto the actin and hold that continued contraction, okay? And this is called smooth muscle tone. And it makes ATP by aerobic respiration. So we're not really looking at anaerobic here at all, primarily aerobic. In terms of special features also, so the special features of smooth muscle is its response to stretch, okay? And it actually has a stretch relaxation response. So it will respond to stretch briefly, then adapts to new length. And you've experienced this because around your stomach is smooth muscle. So different examples I can give is think of a time when you're going to eat a lot of food. So for some people who are in Ontario, that was this past weekend. We just had our Thanksgiving holiday. Traditionally, you get together with family. If you're not vegetarian or vegan, you cook a turkey with potatoes and salad and stuffing and gravy, and it's lots of food, and you have dessert, and you eat, and afterwards you go, oh, I'm so full. But your stomach has been able to expand, and then the muscles around the stomach, the smooth muscle fibers around the stomach, have been able to adapt. And if you don't get Thanksgiving, well, there's other celebrations, right? So think of the celebration of Holi for Hindus or of Ramadan for Muslims, right? After Ramadan is fasting for a month during daylight hours. And then at the end, you break Ramadan with a big feast. Now, you can also eat when it's not daylight hours, which brings up an interesting point. Like, I'd be interested to talk to Muslims about their experience when Ramadan occurs during the summer months and they live in northern Canada, such as the Yukon or Whitehorse or Nunavut or Iqaluit, right? When you have 20 hours of daylight a day, that could be a long time to go without food. But the whole idea is your stomach can adapt. The muscle fibers around your stomach can adapt to that new length, okay? And they retain the ability to contract on demand, which you want because when your stomach's stretched, the stomach muscle, the muscles around the stomach, I'm not talking about your rectus abdominis, like, oh, look at my six pack. I'm talking about the muscles around your stomach. They still need to contract around the stomach to mix up the food so they can then go into the small intestine. Okay, so they can still contract on demand. And really, this enables the organ, such as stomach. What's another one? Or bladder to temporarily store contents. And this is a good thing because as your bladder, your urinary bladder fills up with urine, when you need to go then to the bathroom to dispose of that urine, you want the bladder to contract to get it out. So length and tension changes can contract when between half and twice the resting length. Okay, and it allows the organ to have huge volume changes without becoming flabby when relaxed. Right? So the muscle, the stomach and the bladder can still expand and then empty the contents and not become kind of flabby. They still have a good muscle tone in those organs. Okay, so I'm at 250. So what we're going to do is we're going to take a break right now. So we talked about smooth muscle contraction, not striated. Nervous stimulation may cause them to contract or not, be inhibited from contracting. We have basically autonomic neurons, and primarily the calcium will come from outside of the cell or maybe the sarcoplasmic reticulum. Doesn't bind to troponin. There is no troponin. Binds to calmodulin, which activates the kinase, which then allows for energy transfer from the ATP to the myosin head, and then contraction to go. And then we have to actively, the calcium will loosen from the calmodulin, and we actively pump it back out of the cell or to the sarcoplasmic reticulum. So let's move on with here. So there are different types of smooth muscle fibers, and the type will depend on basically it varies in different organs by three things. First off, fiber arrangement and organization. Okay, so depending on how the fibers are arranged. Second thing, innervation, how the nerve tissue is feeding it. And then responsiveness to various stimuli. So how does it respond when it's given a stimulus? Does it contract or is it inhibited from contracting? So that's the different variations, but in terms of actual categories, you have either unitary or multi-unit. And unitary will and multi-unit are basically two different categories, and it depends on where they are and what they do. So let's start with unitary. Unitary. So it is more common. Okay. It exists in the walls of hollow organs, except the heart. Oh, sorry about that. It is arranged in opposing sheets, and it's innervated, which means the nervous tissue that sends its signals by varicosities of autonomic nerve fibers. Okay, so it has common rhythmic, spontaneous action potentials. So common rhythmic, spontaneous action potentials. The action potentials, the signals to contract could be, seems like out of nowhere, spontaneous. It's not like they got a signal. You don't have to have the signal. Rhythmic, they're going to contract together and it's common. Now, multi-unit, obviously if unitary is more common, multi-unit are going to be more rare. Locations could be in the lung airways, in the erector pili muscles, and those are for hair follicles. So think if you're cold and your hair stands on end, or if you're nervous or you're scared, the hair stands on end, right? Those are the erector pili muscles. And the muscles to adjust pupil size. So the pupils, the openings in your eyeballs that let the light go into the eye, hit the back of the retina, that pupil size is controlled by multi-unit smooth muscle cells, and it's automatic based on exposure to light. So if you're in a darker room, they open the pupil up more. If you're out in the bright sunshine, they close the pupil down. So the multi-unit, similar to skeletal muscle in the sense of they are independent. They contract independently. They have a rich supply of nerve endings, and they respond to neural stimuli. Whereas unitary may respond to the environmental stimuli, or they just may contract. However, the one thing about multi-unit is they are not voluntary. I mean, you can make your pupils dilate, open up by going into a dark room, or constrict by going into a light room, but it's not as if you could just sit there and go, okay, dilate, constrict, to the muscles within your pupils. So that's basically smooth muscle cells. Now, where we're going to end off with is the developmental aspects of muscle and throughout the lifespan. So we're going to go from birth to old age within a matter of a few slides, 4 slides actually. So in terms of all muscle cells, cardiac, smooth, skeletal, all muscle tissue develops from embryonic myoblasts. Okay, so all muscle cells come from embryonic myoblasts, a specialized type of muscle tissue. I mean, really, when the sperm and the egg join and you have conception there, you have one cell, then two cells, and then four cells, and then 8, and then 16, and then 32. So it divides, divides, divides. But at some point, there becomes specialization. So you start to get these myoblasts, which are really kind of like the starter muscle cells. Okay, multinucleated skeletal muscle cells formed by fusion of many myoblasts. Okay, so you get a whole bunch of myoblasts which will join up together to make one skeletal muscle cell. The growth factor stimulates clustering of acetylcholine, ACH is acetylcholine receptors at neuromuscular junctions. So where those nerve endings come in to join with the skeletal muscle cells, you have growth factor, which will stimulate the acetylcholine receptors at the muscle cell. Okay, so you get a few differences here, and this is where we will talk about cardiac tissue in terms of cardiac and smooth muscle. Ooh, smooth muscle myoblasts do not fuse. Okay, they do develop gap junctions. So as we've already identified, the smooth muscle cells join in a rhythmic contraction to help promote, say, the passage of food along the small intestine or mixing of food in the stomach. Okay, and cardiac muscle cells start pumping when the embryo is 3 weeks old. Okay, so as when you have conception, now you have a unique set of DNA there, and within 21 days, 22 days, the actual muscle cells are going. The muscle cells of the heart will start contracting, and they keep contracting until either you're dead or you get a heart transplant. Myoblasts, like skeletal. Sorry, you will also get myoblasts like skeletal muscle satellite cells, which have limited regenerative ability. So what does this mean? Well, when a muscle cell is damaged, you have these satellite cells, and these satellite cells, almost think of them as if they're dormant, they're asleep. Skeletal muscle cell is damaged, or your bicep brachii muscle cell is damaged from a workout or injury, and hopefully it's not damaged so much that you just get scar tissue. Let's say it's damaged to the point it can be still repaired. Well, these satellite cells will wake up, they'll activate, they will multiply, and then some of them will go to repair the muscle cell, and some of them will fall back asleep. They'll become dormant again. And it becomes interesting because, for example, there's a condition called Duchenne's dystrophy. And Duchenne's dystrophy is a genetic disorder where basically the proteins within the cells, within the skeletal muscles, make the muscles very fragile. So the muscles break down with less stress. I mean, when you go to exercise, you are breaking down your skeletal muscle cells. You are damaging them, but not so much that they can't be repaired. And they get repaired and they develop more mitochondria and more capillarization, and they get bigger and stronger. They grow myofibrils. Well, with Duchenne's dystrophy, the muscles become damaged so often that the muscles start to be infiltrated with scar tissue. And sadly, most of these people die in their early 20s from respiratory failure. The diaphragm, the main muscle for breathing, becomes so non-functional that they just can't breathe. Well, one of the theories is that maybe because the muscle cells become damaged easier, they basically run out of satellite cells. It's like they deplete their pool of satellite cells and they go, well, the body goes, well, I have to replace this tissue with something, so I replace it with scar tissue. So there's one theory about Duchenne's dystrophy. Now we're going to make some more notes on this page. I'm just going to have to clear this to make space for it. So cardiac myocytes. So these are heart muscle cells, can divide at a modest rate. It means you can get heart tissue to regenerate somewhat, but injured heart muscle is mainly replaced with connective tissue. So what does this mean? We'll get into this with the heart suction after the midterm. But a heart attack is when one of the arteries feeding heart muscle is blocked. And so that heart muscle is no longer receiving oxygen or nutrients, and it can starve and it can die. Well, if you have enough damage, and it doesn't have to be a lot of damage, if you have enough damage, if those heart muscle cells in that one area die, the body will replace it with scar tissue, with connective tissue. And the problem is that connective tissue cannot contract. And so now the heart becomes less efficient and can promote basically heart failure. Now, the good thing is smooth muscle can regenerate throughout your lifespan. Okay, so you can keep regenerating smooth muscle cells. And cardiac and skeletal muscle can lengthen and thicken in a growing child. In adults, this leads to what we call hypertrophy, right? So hypertrophy is when the muscle cells get bigger themselves. So that's basically some of the development of your muscle cells throughout your lifespan. Now, when we're talking about actual coordinated control, and yeah, it's a picture of my two kids from actually 7 years ago. So muscular development. So we're talking about human growth. And if you have done a degree in, say, physiotherapy or kinesiology, or you go on to a degree, you will take a course specializing in human development, motor development. So muscular development in infants reflects neuromuscular coordination. Okay, so should any of you choose to become parents, or maybe it was an accident, he slept, she fell, and 9 months later, there we go. You need to realize that there is a pattern of coordination development within children. And if you're the parent with a child and your child's 18 months and they still haven't walked, don't panic yet, right? Because there's a window. It's not like at this age, this child can do this. It's a window of development coordination. And in fact, sometimes when babies and children develop too quickly, it can lead to other problems. So, not to cause panic there, but you keep an eye on it, you go to a physician, they compare their development to what the charts are, and they say, yep, yeah, well, it's still within the window, so we're fine. Okay, so development occurs basically from head to toe, okay, and proximal to distal. So what does that mean? Well, when babies are first born, I've been through it 4 times, got 4 kids, you take them home and they're useless. They eat and they sleep and they fill their diaper, and that's about it. But as the child can perform things, well, what's the first thing it does? It can lift up its head and control its head awkwardly. Looks like a drunken sailor. And then it can move its upper body, right? So you give them tummy time, and in tummy time, they can lift up their head. They're lying on and they can lift up their head and come back down. And then they can use their upper arms and then their lower arms, right? Because it goes head to toe, proximal to distal. So even if they can control their arms, their elbow and their shoulder, they're still very awkward with their hands because that's distal. That's the furthest away from the midline of their body. Peak natural Natural neural control occurs by adolescence. Okay, so by puberty, by adolescence, like teenage years, peak natural neural control of the body, that's when it's at its best. Now, one thing to keep in mind, athletics and training can continue to improve neuromuscular control, right? So if we take a child, or let's say an adolescent, someone in their teenage years, and we train them purposely, and so I'll give you one example is either in second or third semester, you'll have Greg Eskigen as one of your instructors. He usually teaches nutrition in the third semester. You can talk to him because he does quite a bit of work still with hockey players in terms of training and development. And I've worked with athletes who have been in adolescence, and you work on purposeful skills to increase and improve their neuromuscular coordination. But having said that, if you're not purposeful about it, then after your teenage years, your coordination starts to come down. And you have to realize this also, that if you're working with an 8-year-old athlete, their coordination will not be as good as the 12-year-old athlete. Because I realize that there will be students in this program who want to work with athletes. Well, guess what? When you want to work with athletes, you're probably going to start working with children at some point. In terms of developmental aspects of muscle fiber, female, so we're talking biological female skeletal muscle is really 36% of body mass on average, right? Male skeletal muscle is about 42% of body mass on average. Well, why in general? Well, I mean, always without intervention. So let's take away in general, biological males have more testosterone. Almost fit it in there. Right? So this is a anabolic hormone which will promote the development of muscle fibers. So males have greater ability to enlarge muscle fibers. Now, having said that, females can develop their muscle fibers also. And in fact, some terrific feats of strength and ability I've seen in female athletes. So for example, one of the few times I've seen someone perform wide grip body weight chin-ups, full range of motion, 15 of them consecutively. So holding onto a bar, supporting the body weight, pulling themselves all the way up and all the way back down 15 times was by a female. Okay, not even a lot of guys can do that. So having said that, but everything else being the same, biological males will have greater muscle mass than biological females, but that can be manipulated and trained. In terms of the muscle mass itself, per unit of muscle mass, body strength is the same. So if you take, say, 100 grams of muscle fiber from a male, biological male, 100 grams of muscle fiber from biological female, they can produce the same, and there's the same muscle fiber types, they can produce the same amount of force with the same stimulation. Aging. It's not for the faint of heart. So, aging muscles. And this becomes relevant for your parents and for your grandparents. So with age, dum, dum, bum, connective tissue increases and muscle fiber decreases. By age 30, loss of muscle mass, which is called sarcopenia, begins. And I know what you're all thinking. 30 is so old, is so far away. It's going to come for you quick and sneak up on you like a mugger in a back alley and rob you blind. However, there is good news, and this is the purpose of this program. Regular exercise, not eggs and fries exercise, reverses sarcopenia. Okay? So we have studies, we have decades and decades of studies which show that people, if they have not been regularly exercising, but they start in their 20s, in their 30s, in their 40s, in their 50s, in their 60s, in their 70s, in their 80s, even in their 90s, you take people who are 90 years old and give them a properly designed resistance training program, it will reverse sarcopenia. It will assist in increasing their muscle mass. One other concern, though, is atherosclerosis. So what is this? This is basically blockage by plaque of blood vessels. You know, this is what contributes to heart attacks. So it may lead to what they call intermittent claudication, which is basically limping, okay? And it could be severe pain in leg muscles. So what does this mean? So as we age, depending on diet, genetics, lifestyle, you can have plaque buildup within your blood vessels. Now, the good thing is that exercise can diminish this, like keep it from happening as quickly. And we have evidence that can even reverse it. But if you're pick the wrong genes and you have a poor lifestyle and you're not regularly exercising, you will have promotion of plaque within blood vessels. And what this does is this restricts the amount of blood going to the active tissues so that you go for a walk, you're 50 years old, you go for a walk, you have atherosclerosis in the arteries feeding the leg muscles, and basically you will get leg pain because they're basically not getting enough oxygen. Even though your heart and lungs feel fine, you're getting leg pain because they're not getting enough oxygen and nutrients in order to support them in that activity. So this goes back to a point where we've mentioned this before. If you are not regularly active within this program, if you're not regularly exercising, start now. If you require assistance and you want feedback in terms of how do I do this, talk to one of your instructors, either to myself or David or Marty. Or I'm trying to think, depending on who you have for fitness instructor leadership, it would be either Ellie, Julia, or Candace, and we're able to point you in resources and give you feedback and assistance to start that. So we'll end the annotation and we are going to stop the recording.